Good evening, everyone. Welcome to Science Distilled. This evening's event is themed Advances in Cancer Research, and we are so glad that you decided to spend the evening with us. This month marks 5 delightful years of doing Science Distilled event. It has been both enlightening and fun, and I'm so glad that you all have been part of it. This evening, we are fortunate to have 3 experts in cancer research in multiple time zones. And they're going to be sharing just the state of the knowledge of this, this topic. So I'm really excited to hear what they have to say. Before we go any further, we want to acknowledge the original and continued sovereignty of Native nations in our region and everywhere. We acknowledge that the places we conduct science have deep ancestral ties to Indigenous peoples, and we recognize that these communities continue to thrive today. If you are here on Zoom or if you are watching on YouTube, we welcome you to type your questions into the chat or the comment box at any time. When it is time for Q&A, we will call on audience members to unmute themselves and ask the questions that they submitted through the chat. Otherwise, we just ask that you keep your mic on mute. Science Distilled is co-hosted by the Terry Lee Wells Discovery Museum in Reno and the Desert Research Institute. DRI is the environmental research arm of the Nevada System of Higher Education, and we are really privileged to work with the Discovery on this. I'd like to now introduce Patrick Turner to tell us a little bit about the Discovery.
Good evening, everyone. Welcome to another installment of Science Distilled. I'm happy to see so many folks joining us, and I see some familiar names in the list, which is great. For those of you who aren't familiar with the Discovery, we're a 67,000 square foot hands-on science center located right in downtown Reno. If you haven't visited, we invite you to do so. Beginning the 12th of June, we go back to 7-day-a-week operations. So there's plenty of opportunity to come visit the museum and explore all kinds of different aspects of science. One other note before we proceed with tonight's event, we normally host this event at the Patagonia store in downtown Reno. And so we want to definitely want to acknowledge their continued support of this series, and we usually pour beer from Imbibe Custom Brews, so we want to thank them as well, and we look forward to getting back to hosting events downtown and pouring those beers from Imbibe very soon.
Thank you so much, Patrick. This event is very exciting because DRI is now home to the Center for Genomic Medicine, which is an evolution of the partnership between DRI and Renown Health. And all of our researchers tonight are affiliated with that collaboration. Our first speaker is Maria Laura Santomo. She received her master's degree in molecular biotechnology from the University of Turin in Italy, and she's now enrolled in a PhD program in biomedical science and oncology at Candiolo Cancer Center in Italy. So she's here in the US as a visiting scholar doing PhD research at DRA Center for Genomic Medicine, and she is sitting in for Kelsey Berry, who was previously advertised as our first speaker. And with that, I will get Maria Laura's slides going, and she will come on and let us present.
Yes, okay. Good evening, everybody. Thanks, Megan. I'm very happy and grateful to be here. So I want to thank you, the organizer, for involving me in this event. And so today I'd like to introduce you to clinical trial. And so in the next few minutes, let's learn to understand much more about clinical trial. That is not always so obvious, and identify how your participation in science helps and know exactly what's going on with your care. So next, please. Okay, as Megan told before, I'm a PhD student in biomedical science and oncology. And I have got my master's degree in molecular biotechnology at the University of Turin in Italy. And now I decide to stay in the States and joining the Center for Genomic Medicine, a program derived from an effort between DRI and Renault to continue and finish my PhD program, which is focused on translational medicine in cancer. Next. Okay, so to begin, imagine you are in a busy hospital looking for a department and you don't know which way to go. That feeling can be very similar to deciding if you want to join a trial and what kind. Next. It may happen that you are asked to be part of a clinical study, which may be an observational study or a clinical trial. First off, both of them use voluntary human participants and are really important in improving healthcare. But observational studies usually require very little effort for you, as the investigators are simply observing you and trying to gather information. There is no treatment or drug being tested. On the other hand, clinical trials are more involved. There is something being tested for efficacy, such as a drug, a diagnostic test, or a clinical procedure. So next. So before testing, what I want to tell you is that before testing a new drug on humans, extensive preclinical studies are done using multiple tissue samples and at least 4 different animal model. Usually 2 of them are rodents and 2 are superior species, which one is a non-human primate. So the goal is to provide information on efficacy, toxicity, and to decide if a drug is ready for clinical trial. And the entire process takes at least 10-12 years of average. And so it is also, it is a highly regulated and controlled by Food and Drug Administration. So only if the results are promising and only after FDA approval, the drug will advance to clinical trials, where its dosage, effectiveness, and safety in human will be determined before the drugs are approved and available to the general public. Next. So why should you participate in a clinical trial? Next. Let's imagine this gentleman, Bob. He was just diagnosed with diabetes. Now he is concerned and has a lot of questions. Is the doctor sure it's diabetes? How does the doctor know what drugs to give him and how much, and if it's safe? Will he have any side effect from the drug? All of these are very important questions. And now, next. from the doctor's perspective. Let's answer his question. The easy answer is evidence-based medicine. All the doctor choice are based on evidence, and this evidence comes from our research and clinical trials, so we need participants to make sure the clinical trials can continue. Next. Thank you. So before we move on, I just want to quickly recap why you should participate if you feel comfortable to a clinical trial. I think you have to participate for you, for a community, and for medical advance, because all the drugs and tools we have today to prevent, detect, or treat disease are thanks to participants in clinical trial. And the clinical trial is the heart of all medical advance. So we can't make progress without volunteer. We need healthy volunteer to help others and to contribute to moving forward science, as well as we need volunteer with a particular illness or disease, again, to help others, but also to possibly receive a newest treatment that are not yet available and to the general public, but that can be much more effective than those available as a standard of care. So if the doctor proposes to you a clinical trial, feel lucky, you have the possibility to receive today the treatment of tomorrow. Next. So if you join the trial, you should feel comfortable asking the doctor or researcher as many questions as you need. So here I report just a screenshot from NIH, which is a very helpful website. They have a prepared list of many questions you could ask. The site also has a number of other pages with everything about clinical trials, including how to find that. So, but of all of them, these 5 that I report are the big 5 categories of questions that you should consider asking. So first, ask about the study. And you have also to ask what are the risks of the study and if there are any possible benefits. You have to ask what kind of participation do they need. You have to also to ask if there are some effect for your daily life. And lastly, will it cost you? Most early studies should not cost you out of pocket. If it is a later phase study, it may go through your insurance, so please ask them. Next. Okay, great. That was a lot of information, I know. Let's make a moment to go back to the busy hospital hallways we started with. So what if instead of clinical study, we were interested in going towards sample donation? Next. Okay, there are multiple ways that you can do this. Researchers are always looking for additional samples to work with to help produce better drugs and tests for us in the future. You can donate anything from body fluid like saliva, blood, and sample. And from my side, I can tell you that it's very powerful. My PhD project is based on plasma sample from patients with gastric cancer and from healthy volunteer. And it's very important to understand the pathology, to understand why some patients can respond to the therapy and why some other patients can't respond to the therapy. So it's very helpful for clinicians to promptly manage the disease. Okay, you can also donate other part of some other tissue. For example, if you were having a liver biopsy, if your doctor was working with a study, they might ask if it is okay for you to take an additional piece, very tiny, for research. And this happen also can help build biobank, which we will talk about this on the next slide. And the last things that I want to say is that regarding the end of life donation, if you were in an accident or passed away unexpectedly, it can be very helpful to know what you want with your body. So donating to living donor is always a consideration, but if that is not an option because your organs are not healthy enough, donating to science is too. So next, please. So where are your samples going? What is a biobank? Next one. Okay, here I'm showing you what happens to your sample when you donate to science. Sample travel to appropriate lab for research. And the picture that you can see on the left, in particular, is a great example of small biobank. Your tissue will be carefully cared for, often mixed stored in ultra-low temperature for a long period of time to preserve them until the usage. What is stored is your sample, but also your clinical data. But the clinical data include sex, age, disease, but what I want to say you is that not include your name. We use a specific code number, so your name is nowhere. Next one, please. This is another example, but is a great example of a much larger biobank based in the United Kingdom. So this UK bank is a worldwide powerful tool to develop new drugs and move forward to better understand the disease, and so on new approach and prevention. As you can see, they have a massive and highly organized structure, and so represent a great example of a community effort to donate tissue sample to science to promote the research and to improve health, human health. Next one. Lastly, speaking of community program, I want to introduce you to our very own program based right here in Pino. We are extremely lucky to be the home base for the Health in Nevada project. This is a unique genetic study that is working to answer health questions for the entire community. Last one. What makes this project unique is that it's the largest community-based project populational study in the world. The purpose is to offering genetic testing to any interested Nevadian and to give them more information about the healthy and the genetic risk. So one important thing is that it's free to participate in. What they ask for is just a sample of your saliva. So your DNA is taken from the sample and sequence, and the genetic information is shared with you and also used as a model to answer health questions for communities across the country. So up to now, it is possible to make a screening of certain type of cancer genetic marker like breast, ovarian, and Lynch syndrome. And it is very, very important from one hand because for the researcher to understand why certain people get cancer and how genetics, environment, and social factors impact our health. But on the other hand, it's very important for you, for your early prevention, because it's well known to be the most effective line of intervention, even more than standard of care. Okay, so I finished my presentation, and I leave the stage to Sarah.
Thank you so much, Maria Laura. I am still learning to use the mute button. That was wonderful. So we really appreciate you stepping in, sharing your knowledge, giving us a really strong baseline of what clinical trials are and the other ways that we begin to understand the varieties of cancers that are out there and why they're prevalent in the community. So Maria Laura just talked about the importance of biobanks and how to approach a clinical trial to get all the information that you need. Our next speaker, Dr. Sarah Gandalfi, is going to build on that by explaining in more depth the details of a clinical trial and its impact on patient care. So we are really fortunate. Dr. Gandalfi is located, she is currently in Helsinki, Finland. She is an MD, PhD, and a postdoctoral research fellow at the University of Helsinki. Dr. Gandalfi worked for 5 years at the Dana-Farber Cancer Institute at Harvard Medical School in Boston. Their research studies are focused on better understanding how blood cancers evade the immune system. All of her work is focused on improving immunotherapy efficacy. So with that, I will turn the microphone over to Sarah.
Great. Thank you so much, Megan, for the nice introduction. And yeah, I'm going to talk to you about how to fight cancer as a team. So as Megan nicely already mentioned, here a bit of background and my journey. So everything started in Italy, where I was born and raised and where I studied medicine and where I was working as a resident physician. And I was working in clinical trials in bone marrow transplant and blood cancer. But that wasn't enough for me. So I decided to move to the United States, specifically to Boston, where I worked at the Dana-Farber for 5 years, trying to figure out how the immune system can be leveraged to fight the patient's own cancer and how to improve the immune therapies and try to, learn better how cancer can evade our own immune system and grow. And the same project led me to land here in the very northern Finland, where I'm still working on studying how cancer cell becomes smart and can hijack the immune system in order to grow and how we can fight that. So I'm basically working on immune therapy, but what's all this immunotherapy all about? Well, immune therapy has been work in progress for a very long time. So the whole story starts back at the end of the 1940s, and it's when basically, so from the observation of mouse models, so scientists observed that mice could recover from leukemia after very strong radiation and the administration of bone marrow from other healthy mice. So in between that and 1969, a lot of research allowed scientists and physicians to treat the very first immunodeficiency with a bone marrow transplant from a healthy donor. And in between then and the current times, a lot of research, a lot of new discoveries, as well as a lot of failures allowed us to land to the present days, in which we're not only able to give bone marrow transplant to hematologic patients, but we wanted to expand the immunotherapies to other indications, to other diseases. And we are basically now able to create antibodies that are specifically targeting the immune cells or the cancer cells to wake up the immune system and help the patient fight cancer, as well as we are able to engineer the immune system cells so they can target more specifically the cancer cells and can still patrol the body in order to identify new cancer cells arising and fight them back. And we are also able to develop these so-called cancer vaccines. So a lot of research, a lot of novel discoveries that are still ongoing. And actually, to make this happen, so to go from the so-called bench to the bedside is a very long journey that requires a teamwork. It requires scientists for the preclinical phase, in which there are the very early days of a drug development. That could be a new medication or a targeted therapy or immunotherapy. So there's the drug discovery, the formulation, and how it can be better delivered to the patient with the lowest toxicity level. And once a drug goes through all these preclinical steps, the nerds just, give the floor to physicians so they can translate these discoveries to the clinical side. And that's when clinical trials happen. So clinical trials are a very safe process in which patients can get either the best available treatment or the best available treatment and even probably better with a new drug. But this new drug has to go through very strict steps before getting to the clinic and during the clinical trials. So clinical trials are made of 4 phases. The first phase answers the question if the drug is safe and what dose should be used. And it's tested in a very small population. It can be tested in either healthy donor, but for cancer medication, it's a smaller population of cancer patients who have this new drug as the new hope to try and get better. So it tests the safety, the dosage, starting very low dose and then expanding and ramping up the dose. Then once the drug goes through phase 1 and passes the phase 1, phase 2 happens. So it's a slightly larger population, and it tests the efficacy of the drug. So it answers the question, does this actually work? And are we seeing any negative effects? Are there any toxicities that need to be addressed? And how can we get around that? So it's basically efficacy and side effect kind of study. Once it goes through phase 2, then we can move forward to a much larger phase 3 clinical trial. It's usually much larger population, thousands of patients. Usually there's a comparison between what's the best available treatment. So there's no such thing as being a guinea pig in a clinical trial. This is a very common misconception. All these drugs are very well tested, and at the worst case scenario, the patient gets the best available treatment currently available, or even better. So in phase 3 trial, we fine-tune the dose, we find the perfect dose for that patient population, and we try to improve the treatment with combinations. So it's basically wider population, and it also tests, fine-tunes also the effectiveness. Once it passes the phase 3, the drug goes for FDA approval. In that moment, when FDA approves the drugs, it becomes available to whoever has the indication that was studied for. And then we move forward to the phase 4, in which we ask ourselves, what else can we use this for? Or can we see like some long-term side effects or some long-term effects of the drug? And this is the post-marketing drug testing, and it can also, help expanding the indication. But throughout all this very long and super controlled process, which also allows the patients to get the best available treatment, novel options, help science, as well as being super controlled by a whole clinical team, the nerds are still working, and there's translational research ongoing all throughout the time. So scientists like me, I'm a translational physician. So I work both with patients and at the bench in order to identify while patients are in clinical trials, but even before they get into clinical trials, tiny little research things that help us identifying and predict mechanism of resistance to a specific medication, or testing in the laboratory what would be the best combination with other medication. And then in the clinical phases, we sneak in patient samples, and we try to study how the cells behave, like patient cells behave in the presence of the medication, and try to figure out what are the patients that have the most chances to respond to the specific treatment, and what patient might already develop some resistance, so we can even improve the treatment by adding some other medication or other therapeutic options. So in all this process, our team needs some help from the other team, which are patients and healthy donor. For the preclinical phase, we're always happy to receive patients or healthy donor samples so we can study the drugs before they get to the market, also in relation to like microenvironment, or as I said before, testing other combination. And comparing what went wrong between patient samples and healthy donor, and we can find out a lot of novel genetic hits that might become give us a new ideas for novel treatments. And in clinical trials, we need clinical trial participation. So having patients getting to know that clinical trials are available and what are the options, and that they are open for patients to, of course, to access if they have the requirements for the clinical trials. And your doctors can help you and guide you through the clinical trials available in order to find the best one for the patients. So how do we connect this huge research world with the bench side, and how we make this translational thing happen? This is what we do here in Helsinki, but it's something that a lot of research groups are working on. We are trying to move towards a personalized approach. It's still a long way. It still needs a lot of testing before it becomes a reality. And that's why we need patient samples, healthy donor samples, because the more we study, the more we learn, the more this can become a reality. So what we do is that we take patient samples and we treat them with these novel, amazing immunotherapies with specific cells that carry some receptor that recognize cancer, or these novel antibodies, and we combine them with other drugs, and we try to figure out what are the drugs that in that patient population, in that specific patient, can elicit the best response. And then we study the genetics of these cells, we study the mutation, we study how they interact with the new drug, and we try to profile them and to integrate this data with the patient's clinical data, so we can fine-tune the treatment and identify the best treatment for that specific person. So that's our dream, that's what we want to do, but as I said, still a long way to get there. So I hope this was a nice overview of what we do in translational research, and I want to thank you. And I also say it in Italian, grazie, and in Finnish, kiitos, for listening, and I will be happy to take any questions. And I leave the floor before taking questions to Ricardo.
Thank you so much, Sarah. I appreciate how much time you have spent really finessing your skill in translational medicine, not only between the research and the clinic, but helping us generalists understand the specialty that you have. So I appreciate that skill that you have developed very much. And in the Q&A later, we will explore a little bit more this team-based approach that you mentioned and why it is so important that community-based studies are taking place. So thank you so much for sharing.
Thank you.
So Sarah just shared a lot of information about clinical trials. She walked us through the process and really unpacked it for us, because if you're like me, you didn't know kind of what happened beyond the door of the hospital and what it meant to really participate in one. So she really unpacked that for us. And our next speaker, Dr. Ricardo Panela, is going to explore what exactly standard of care means. It's a term we hear a lot, but what is behind that? What does it mean from a researcher and clinician point of view? And then he's going to build on that by looking at what it means to move from standard of care to standard of cure. And that's going to involve taking advantage of both clinical trials and biobanks. So Dr. Ricardo Panela is an assistant professor in cancer biology and genetics at the Center for Genomic Medicine here at the Desert Research Institute. He worked for 6 years at the Beth Israel Deaconess Medical Center Cancer Institute and Harvard Medical School Cancer Center. And we are very fortunate that he came to join us in Reno. So with that, I will turn the mic over to Ricardo and take it away.
Good evening, everyone. Let me share my screen so I can show you my presentation. and walk you through it. Okay, that should work. So first of all, I want to thank you, DRI and Discovery, and thank you, Megan and Patrick, and all the people that are behind this Science Distilled series. There is a fantastic series that it really brings science close to our community, and I think this is crucial and it's very important, especially in a period like the one that we are facing, in which science can really help moving forward with the pandemic, for example. And so, as Megan already introduced me a little bit, I want to give you an overview of who I am and where I come from, just to know each other a little bit better. So, sorry for my English, it's not perfect, I'm working on it. I have an accent, I speak with my hand, I'm Italian. So I was born and raised in Italy, where I completed the whole my educational pathway from the master degree in biotechnology to the PhD in oncology. And after my PhD, I realized that was time for me to move over. So I crossed the ocean. With the kayak took a while. It took like several months for me to do that, but eventually I landed in Boston. And I joined the Harvard Medical School, and specifically one of the teaching hospital of the Harvard Medical School. And for that 7 years, almost 7 years in Boston, there was a lot of lab, scientific meeting, lab, scientific meeting, and there was some foliage and some nice hiking involved here and there. And then in the middle of a pandemic, since my life was not disturbed enough from the pandemic like all of our life, I decided to move again. And in September of 2020, I joined DRI, where I'm establishing my own research group. And I have to say, I'm very excited and happy to be here. I found a very interesting scientific community and environment, and Reno, it's treating me very well. So I'm very happy and honored that I can serve DRI and the Nevadian community. So with that said, I will move forward and I'll tell you, I'm interested in translational medicine for my entire career. So as Sarah already mentioned, translational medicine is that part of science that connect the bench side to the bedside. So I really want to do research that can affect patient life in the short amount of time possible. So as per today, the standard of care when we talk about cancer, it's effective and it helps in saving millions of lives every year, but has a lot of room for improvement. So let me explain how does it work. Basically, when patient X arrive at the hospital and they receive a cancer diagnosis, they are treated with different therapies, because for each kind of cancer today, we have several therapies that can work or cannot work, because each cancer, it's not one disease. So for example, for lung cancer, we have a lot of subtype of the same general disease, lung cancer, and each subtype can be responsive to a therapy or to another therapy. And initially, clinician doesn't have a really a good idea or a strong clue to understand if for patient X, therapy 1 is better than therapy 28 or is better than therapy 54. So what happened is that they start treating the patient, and this therapy may or may not work. And if it's not working, the patient go through another cycle of a different therapy until the clinician find a therapy that it's able to reduce cancer. So when a cancer is responsive to a therapy, we are applying a pressure, a selective pressure to this cancer. And when we apply a selective pressure to any biological system, the biological system tends to find a way to escape that pressure that we are putting on it. And in cancer, this is called resistance. So at certain point, the cancer stop responding to the therapy that was effective till a week before, and it become resistant to that therapy. And when resistance occurs is where we draw the line between the first line therapy and the second line therapy. And when a patient develops resistance, the standard of care as per today, it's very similar in the second line of therapy to what the patient experienced in the first line of therapy. And so different target combinatory immunotherapy are tried. Some of them will work, some of them will not. So if we try a therapy that it's not working, we have to try a second one, a third one, until we find the therapy that it's working and it's effective on that cancer, and we can progress and bring the patient to a final stage of cancer-free, to a cure. So this approach, as I told you before, it's saving millions of lives every year, and it's a very potent approach. But as you can see, there is a lot of back and forth. Trying all these different therapy is not ideal. The patient, it requires time, and for certain type of cancer, especially the most aggressive types, time is a factor, and we have to find a way to reduce the time from the diagnosis to the cancer-free stage. And so all these back and forth are not ideal. And on top of that, every time that we propose a therapy to a patient, and the therapy is not effective on the patient tumor, we still have all the side effects of that therapy that in some case can be pretty severe. And so trying multiple therapy also has a problem in terms that we are built up on side effect, and some of these are pretty severe, and the patient can go through serious distress. So it's an approach that it's working, but it's not ideal. So using the idea of personalized medicine that Sarah just introduced to all of us, we are moving toward a much more advanced way of treating cancer. That is what we call the avatar model. The avatar model that I'm going to explain you in a second, it's very effective, but it's extremely costly and very hard from a technical point of view to achieve. So it's an approach that few centers worldwide are proposing to their patient. And but I mean, give us a good idea of how fast we can move forward. So again, patient X arrived at the hospital. She got a cancer diagnosis, and the patient started the first line therapy like a normal standard of care. So, but when the patient is diagnosed with cancer, some of the patient cells from their tumor are harvested and they are implanted in a mouse. So now we have an avatar mouse that it's not carrying a generic lung cancer, but is carrying the exact lung cancer of patient X. And while the patient is starting his path toward the cancer-free stage and testing the first-line therapy, we can expand the mice. We can create a cohort of mice or an army of mice, if you prefer. And all the possible therapies that are a possibility for treating that type of cancer can be tested in these mice. And so we can know in advance if therapy 1, 28, or 52 is more effective in curing the specific cancer of Mr. X. And so we can already avoid that back and forth that cost us months, if not years, in the first line of therapy. But more important, we can push this mouse model, keep treating them till they reach resistance. And once that the tumor in the avatar mouse become resistant, we can use that mouse to test all the second line therapy possible for that particular type of cancer and know before the patient develop resistant what is the therapy that will work in patient X when he or she will develop the resistant. And so when that happens in the patient, clinician already know what is the right therapy to prescribe to that specific person. And this allow us to avoid the second round of back and forth and speed up the entire process big times, because we can predict the right therapy, mainly the second line therapy, but also the first line. And we can predict and we can give to the patient the right drugs on our first shot. This is saving so much time and potentially can save thousands, hundreds of thousands of lives. But as I told you, generating these mice, it's costly. Testing all the possible therapies, it's costly. And so this approach from a sustainability point of view, it's not the best one. And it also has some like technical challenges, because not all the tumor are happy to grow in mice. So there are some aspects in this approach that can be employed. So what is our vision? Our vision starts from the taking advantage of human genomic population data. Like Maria Laura and Sarah talk about Biobank, the big Healthy Nevada project. It's a huge biobank in which data, genomic data from patients and from healthy people, for everyone, are stored and analyzed. And we can use this big set of information to understand what are in the human genomic setting the mutation or the combination of mutation that are important for that specific cancer or disease. And we can use this information from human to build mouse model that carry one or multiple mutation that we discover being important because we analyze the human genome. And so these mouse models are representative of all the possible combination that we can find in a human subject with that specific kind of disease or cancer. And so these mice models are a representative animal model of specific disease or cancer. These mice models can be used to test first-line therapy, pushing resistant, and testing second-line therapy. And this has a huge impact because through this mouse model, we can understand what is the right treatment for a cancer that has the green and red mutation, or a cancer that has the red and black mutation. And so when the patient arrives to the hospital and patient X, Y, Z got diagnosed with cancer, if they are part of the Healthy Nevada project, we already know their genetic makeup. If they are not, we can very quickly sequence them and discover their genetic makeup. And then we can go back to our library of mice model and understand what are the therapies that the tumor of patient X will be responsive to, and go directly with that, speeding up the process, the path toward a cancer-free stage and a curative stage big time. And this approach, it's very powerful, and it's our vision to improve big times the patient overall survival, and it's doable only if this big database and this big biobank are keep growing and growing and growing, because the more people and the more participants we have in the Healthy Nevada project, in our case, but in all the biobank, are giving us more information, and more information means more knowledge, and more knowledge for us meaning a better treatment for all the patients. And it's important for you to participate in this kind of biobank and population study from a very selfish point of view, because you will know if you are more prone, or in case something ever happened to you in the future, you know that there is already a personalized treatment for you, and can be beneficial for all the other people in the community that are not part of the biobank, but can still take advantage of the fact that people participate in this study and help us to give you a better cure, a better therapy. With that said, I will stop bothering you with my talk, and I'm happy to take any kind of question.
Ricardo, thank you. I feel very hopeful hearing about the Avatar model and your work with the Healthy Nevada Project. And I'm incredibly grateful that you're here and that you're shared your work with us. To the whole audience, I hope that you came to grasp the power of community-based studies for this very, very important work that all three researchers are involved in, and the power of the work that their entire teams are doing. I'd like to introduce my colleague, Ali Swallow, who is going to facilitate Q&A. If you haven't already, we welcome you to share your questions in the chat. It's all you, Ali.
Thank you, Megan. I appreciate it. The first question we have tonight is from me, actually. When I was watching your presentations today, I was curious, can can you take us to the moment when you decided to dedicate your career to this topic? I would like to know from all three of you, what inspired you to study cancer?
Okay. Hi, can you hear me? Okay. Yes, it's very funny question because for a few years after my bachelor degree in biotechnology, I worked as a violinist because I also had a master's degree in violin. But I have always been intrigued by the world of science. So I decided to move from the center of Italy to the north and to start my master's degree in biotechnology. And there I had a very good professor in genetic of cancer course. So after I met him, I became really fascinated, more than I already was, by the complexity of cancer and its extraordinary ability to evolve in response to therapy and how escape all the mechanisms, all the therapy. So I decided I really want to study cancer. And so I decided to apply for a PhD in oncology for sure. And it's also the reason why I'm here now.
Thank you so much.
Welcome.
Shall we go in the same order? Okay. Mine is a pretty long story. I have two personal events that led me to study cancer and one more professional, multiple professional moments. But I remember the very first time I learned about cancer. One was my grandfather, who passed when I was very little. I was 2. And then when I was 9, my friend's sister, she was 10, and she passed from leukemia. So that's when reality kind of got into my life. I was like, wow, kids can die from cancer. And I think that was the imprinting. And then fast forward, I was 16, I got a appendectomy, and I was like kind of in between pediatric and adult patient. I ended up in an adult ward, and I was surrounded by all these ladies, and there was this young lady, and she told me her story, and she was like, she had this weird pain in her shoulder, and she went to see so many doctors, and no one could figure it out. And then one day, one doctor just talks to her, listens to her, and the only thing he does, he puts his hand on her liver right here, and he found out the tumor, and she was there to get surgery. I was like, I want to be that cool doctor that listens to people and can really help them. It was a benign tumor for that woman. So then that's when probably I decided that I didn't want to be an archaeologist after all, because that was my first thing when I was a kid, pretty nerdy kid, I'm sorry, but I had very nerdy moments, but I wanted to be a doctor. So when I decided when to go to college, I just decided for med school right away. And then while doing my clinical rotations, I ended up in hematology, like randomly. It was like, oh, wow, that's going to be tough. But my mentor, he's awesome. He's a transplant doctor. And that's where I realized that hematology is just the coolest thing ever. You have internal medicine, you have ICU care, you have research. There's everything. And I met so many people, so many lives that we saved, and unfortunately that we lost. And I was like, I don't want to lose any more patients. So that's where I stepped from being a clinician, which I love, and I want to go back to my clinical work as soon as possible, but to also research, because just clinical work is amazing, but to me, it's not enough. I want to do more and better. So I think that was a spark that kind of built up over time. So that's the story.
That's great. Thank you. Ricardo?
My motivation is more nerdy than the ladies. And fortunately, I don't really had any cancer history in my family. So I was not like touched by cancer before start working with that. Cancer came to my family in most recent years when I was already abroad. And so it was not the first reason why I started to do this. But when I was like a biotech student, my first experimental experience was in a lab during my thesis and was on Alzheimer. And it was very fascinating, and the entire lab environment was very fascinating to me. And then studying molecular biology during the last year of my master degree, I stood up on cancer. And cancer is so complex, so complicated, and I love challenge so much that I told to myself, oh, Rick, you have to pick this up. This is the most difficult challenge that you can face if you stay in science. You need this to challenge you and to solve a problem that a lot of people are working on and no one is solving. I was very arrogant and presumption at that time. And so this is how I start doing cancer. And now I understand that I cannot solve the problem, but I can contribute. Even if I contribute a little bit, it's going to be an important advantage for the scientific community and for people.
Thank you so much. For our first audience question, I am going to turn it over to the president of DRI, Kamud Acharya. Would you like to come on camera and join us to ask your question?
Thank you, Ali. I'd like to thank all three of you for a wonderful presentation. So my question to you is, why is this cancer such a hard disease to treat? How many years are we away from being able to treat cancer? Because we've made so much progress in invention in medical science and everything else, but cancer seems like something that we've never been able to really solve the puzzle. Any thoughts on this, like as to how far away are we from being able to treat, or what is required? I mean, do we need more resources, more people in it, or what is it? What is not helping us to solve this problem?
I can start commenting on that and then having the ladies jump in. So basically, for many years, there was a misunderstanding that cancer is a disease and lung cancer is one disease, like, I don't know, diabetes. This is not true. And lung cancer are a lot of different kind of disease that we call in the same way for the sake of understanding. Okay, but in the reality that we discover is that my cancer is different from my father cancer. And so the treatment for my father cancer may not work on my tumor. So this was really a problem in the beginning on how research and clinician approached the cancer. And now that we are disentangled and understanding that it's a very complex disease, we are going toward this personalized medicine in which we are not treating lung cancer, but this treatment is for Ricardo Panela lung cancer. And it's different from the treatment that we are gonna use for Vincenzo Panela lung cancer. So this was the first step that allow us to treat cancer better and to understand cancer better. And on top of that, we have to think that cancer is a very complex disease with a lot of gene and mutation involved, and it's always in evolution. There are new mutations that appear every day, and the cancer itself change. So as I told you during my meeting, a cancer can respond to a therapy and then change themselves to make that therapy not effective anymore, become resistant. This is a unique characteristic, the adaptation of a living subject, of a living biological subject. So we are facing not just a disease, but a biological system that is constantly evolving and changing. And to treat them, we have to evolve and change our approach toward that disease every day. And I think what we need, it's research are always helpful, and a strong community effort between scientists, collaboration between scientists all around the world, and a big involvement from the community, the people that are part of our community. That's my view.
Okay, maybe I'll just chime in and expand on that. Totally agree with Ricardo's overview. So we've been living with the cancer issue ever since the man was on Earth, because the very first traces of cancer were found in some Egyptian mummies that I had found out from their bones that they had breast cancer or multiple myeloma, because they can be seen by how the bones are structured. So it's been always with us, and it's a disease that evolves in a Darwinian way. So it's it has this selective pressure that we also put with drugs. So it can outsmart what we do. And the more we learn about cancer, the more we understand that probably it's not just the cancer itself, but also the environment, the microenvironment. So our same immune system, our same supporting cells that make the cancer stay alive and protected. So the more we learn, the more we are understanding how to try and tackle cancer from multiple points of view and from multiple directions. And what we are learning from all these amazing genetic studies and from all these patient samples that we get is that there's variability of cancer cells, not only across different patients within the same disease classification. We're doctors, we like classification because we need to understand what to give to whom as a general treatment. But then, as Ricardo nicely pointed out, every single cancer is different, but it's also different within the same person. That's the complexity of treating cancer. And as we don't know yet why cancer X arises in person Y and why a different cancer arises in another person, except from all the risk factors that we all know, there are little things that, why did Jane get breast cancer and Janet got, say, leukemia? Why that? So we don't know yet that, so we cannot go through the source and the roots of cancer. So what we can do now is just treat what we see and try to learn as much as possible, as much as we can. And we are actually blessed to be in these amazing times in which we have so many technologies and probably more to come that will help us to better understand cancer and better try to figure out how to cure it, because the problem is not the treatment, because we can treat cancer. We have amazing treatments. We want to cure it. And that's the challenge. And that's what we hope we will be able to do. I don't know when, but I think we are really heading towards that goal.
Thank you so much, Sarah. Okay, our next question is coming from Ted Hartwell. Ted, if you'd like to unmute yourself and ask your question about the avatar model.
Hi, thank you all of you so much for the presentation. Very informative. Sarah, I appreciate your archaeological background. I started out as an archaeologist at DRI and then ended up managing a radiation monitoring program that's a citizen science program dealing with detection of radioactivity from past nuclear testing. And when I do community presentations on this, I invariably get lots of questions about cancer, and I'm generally able to answer most of them in a very general sense. But and this question maybe is for all of you, but I'm wondering if things like the avatar model are not only promising for developing a database where we can know what treatments will be most effective for certain individuals in the future, but also is there thought being given to things like prevention? I mean, knowing that someone's going to be at a higher risk, and but that they're also they respond well to either certain diets or more exercise than other individuals as a preventative measure for keeping certain cancers. Of course, those are things that are, diet, I guess, and exercise are things we all should do to fend that off. But is there some potential in that area as well?
Thank you, Ted. Very, very nice question. Very interesting. So yes, to give you a short answer, yes, of course. There are a lot of different applications that the avatar model can be useful for. And some of these are also like not the classic chemotherapy, but also combination diet più this drug, diet più the exercise, or how much exercise, how many lipids in the diet, and all this kind of question. The only thing that is not possible to do with the avatar model, it's studying prevention. Because for building the avatar model itself, we need the cancer to be in the patient. So everything that could prevent the cancer, it's something that we can really not study through the avatar model for the limitation of how the animal model itself it's built. But then once that we have a primary tumor that is established, and we can transplant and expand them in mice or rats, at that point we can use these tools for a plethora of different applications that goes far behind the just the normal first line of chemotherapy.
Thank you.
Sure.
Fabulous. We are going to wrap up the event. I want to thank all of our speakers and audience members, and we will leave with two things. The first thing is that I mentioned that we are in multiple time zones. Sarah, do you want to tell us what time it is where you are in Helsinki?
Now, as we speak, is 6:35 in the morning.
It is 6:35. So Sarah got up and started this presentation at 5:30 a.m. So I just want everyone to please show your gratitude for that, as well as our other two speakers who were on the Pacific Coast, 7:30 p.m., obviously. Now a little after 8:40. So thank you so much. We really appreciate this illuminating event. Patrick, is there anything you want to tell us about what's happening at the Discovery coming up?
Thanks, Megan. I think I covered it at the beginning, but I'll just mention again that the museum goes back to 7-day-a-week operations starting this Saturday. We're really excited to have a full week of folks visiting the museum.
Yes, I am too. And I want to thank all of our audience members. We know that life is transitioning from the screen to back in person, and we appreciate you spending your evening with us. And we look forward to seeing you in September for our next Science Distilled called Science from the Sky, scheduled for September 15th. So have a great summer, stay cool, stay hydrated, stay healthy, and we will see you then. Thank you, Maria Laura.
And
Sarah.
fight cancer.
And fight cancer. Get out there and fight. Thank you,
Thank you, everyone. Thank you for having us.
Thank you for this great opportunity, and thank you for this amazing event. It was great. Thank you.
Ciao, ciao.
Bye.